Okay, today we're going to talk about X chromosome inactivation. This process should be familiar to people that have had genetics, but a little reminder won't hurt. And also there's some people that have not had genetics, so this might be new. So this is about dosage compensation. The picture shows karyotypes of normal female and normal male. And the chromosomes are, of course, arranged in pairs in order, with 22 pairs of autosomes and then the sex chromosomes. Now we see that the female has 2 X chromosomes, whereas the male has an X and a Y. So this gives us a biological problem that we need to figure out, because that means that females have twice as many copies of those X chromosome-linked genes, about 1,000 per chromosome. And so we want to equalize the expression of the X so that the male and the female will produce equal amounts of those particular proteins. Here's another female karyotype, and you can see those 2 big old X's on the lower right. So this is going to be an example of monoallelic expression, where we have a dosage compensation system in mammals, whose job it is to equalize the expression of X-linked genes in males versus females. So we don't want to have twice as much transcription of X-linked genes in females as we do in males. Now there's different systems for accomplishing this in different organisms, but in mammals what we do is we choose one X to be active and any extra X's to be inactivated in each cell. Okay, this occurs in early embryonic development, and once a cell chooses a particular X to be inactive, that same X is inactive in all the progeny of the cell. That X is called Xi, and the active one is called Xa. And this is an example of epigenetic regulation. Okay, a reminder about heterochromatin versus euchromatin. Heterochromatin is darkly stained, tightly coiled, and doesn't ever get loose during the cell cycle. So the chromosomes are compact during mitosis anyway, but this particular form of chromatin remains compact throughout the entire cell cycle. The usual kind of chromatin that contains the active genes is the type that changes chromatin state during mitosis and meiosis, whereas the heterochromatin is always darkly staining and tightly coiled, and generally inaccessible to transcription factors. Okay, in mammals, the inactive X is heterochromatic and is very condensed, and as such is visible in the nuclei of females as a bar body or darkly staining visible blob. So as a result of this system, we can actually tolerate extra X chromosomes, even though these are big genes. XX people have 1 bar body, XY people have no bar body. So here's a depiction of what happens during early embryonic development, where the blue chromosome is the paternal one and the red chromosome is the maternal one. Every female zygote has 2 X's, one from each parent. So you have early cell divisions during embryogenesis, but at some point there's a random X chromosome inactivation process that kicks into play. So as a result of this process, each cell chooses one X to be the inactive and one to be the active one, and those are shown as a little dark red or blue blob in these cells, indicating that that particular X has been inactive. So in the nucleus and the cell on the left, the paternal one is active, maternal one is inactive, and the next one is the maternal one is active, etc. So as a result of this process, female mammals are a patchwork of X chromosome inactivations, because half the cells will express the genes from one X and the other will express the genes from the other X. And the bar body is visible as a dark blob in the nuclei of these cells. So the female calico cats are a very visual way of showing what X chromosome inactivation leads to. The female cat has patches of black and orange fur according to which X chromosome is active in those particular cells. Okay, this is a result of 2 alleles of an X-linked gene, which are responsible for the different colors, and only one, of course, is expressed in any cell. So she's genetically overall heterozygous for this, but each cell only expresses one of those genes. Okay, so in terms of the process and how this happens, the inactivation depends on regulatory RNAs called Exist and T6 that are long non-coding RNAs that are produced from a region of the X chromosome, which is called the X inactivation center. Okay, the exist is expressed as a race early on for which one is going to get expressed, but ultimately there's a choice where one of the X chromosomes will express the exist RNA, and the other will express the antisense RNA. So the chromosome that expresses the exist RNA is the one that's going to be chosen, has been chosen, to be the inactive one, and the T6 leads to activation or failure to inactivate that chromosome. So the mechanism here is incredibly complex, but some details are certainly still need to be worked out, but the basics are in place. So here's a chromosome from mouse, which shows where the X chromosome inactivation center is located, and there are a bunch of different RNAs that are produced from this particular region of the chromosome. Okay, the ones that concern us are the exist, shown in green, and the T6, shown on the opposite strand of the same region of DNA, shown in blue. So the exist expression, the chromosome that's going to express the exist, is the one that's going to become the inactive X. Right. So what happens during early embryonic development that initiates this process is the exist is produced from this region, and it coats physically the future inactive X chromosome. The exist RNA does not encode any proteins, and it's quite long, about 17 kilobases in total. And it is made from the X that has been chosen to be inactive. So this is shown just sort of very schematically here in one version. We start off with two X's, both of which have X inactivation center. And the one that is chosen to produce the exist RNA, the RNA continues to be produced, and it essentially coats the entire X chromosome that's going to become inactive. So what it does is it targets proteins and things that are going to cause that inactivation process, targets it specifically to the future inactive X. So this can actually be seen by a microscopic technique called FISH, or fluorescence in situ hybridization. So in this experiment, we're going to see the exist RNA as a cloud which covers the X chromosome. So in this case, the exist RNA is labeled red, and the DNA, generally stained with DAPI, is going to be look blue. So FISH in general is a method for looking at the localization of molecules within cells. Now that could be proteins, could be specific DNA sequences, or specific RNA sequences in this example. So typically, at least in this example, the cells are fixed on a slide, stained with DAPI to stain the DNA. Now DAPI will fluoresce a blue color when it's excited by the correct wavelength of light. Then the chromosomes can be hybridized to something. In this case, we're going to hybridize it to DNA fluorescently labeled DNA probes, or else it could be an antibody if you're looking for a protein. So we need to have a fluorescence microscope that excites the fluorescence molecules at specific wavelengths, and we need to have those molecules tagged with the appropriate different colors according to what we want to see. Okay, in this image, we're seeing the exist RNA in the nucleus. The DAPI is the blue that covers the entire nucleus. And in this case, the exist RNA was probed with a fluorescent compound that fluoresces red. Okay, so, and of course you can see the two different colors. Now in doing this, images have to be captured at the two different colors and then merged to form a final image. So the exist RNA cloud is very visible. So what's going to happen during the process? This is kind of schematic and includes not all of the details. We're going to start off with an initially active X. Then we will express exist from the X inactivation center of that one chromosome. As a result of this coating, we're going to have deacetylation of histones by histone deacetylases. Now remember, acetylation is an mark of active chromatin, and inactive chromatin is deacetylated. We also have a mark of an ubiquitination of histone H2A in response to the PRC, meaning polycomb repressor complex, which includes the methylase, the ubiquitin transferase that does that reaction. Next step, we have methylation, in this case of histone H3 at lysine 27 trimethylation, and that is established by the different polycomb repressor complex 2. Notice that this one gene indicated as an escape gene. This process does inactivate most of the genes on the X, but there are always a few that remain escaped, and people are still trying to figure out how that happens. Finally, we have the inactive X, which is actually some of the histone H2A is substituted by a variant histone, which is called macro 2A. Okay, so as a result of this very elaborate process, the genes on that chromosome are silenced. Okay, so characteristics of the inactive X. We have, of course, low histone acetylation, right? And so we're going to have deacetylases that do that. We're going to have specific repressive chromatin marks installed by histone methyltransferases, including the lyse 27 on histone H3. We're going to have ubiquitination of H2A, and not really shown here, we also have a very compact chromatin structure, which is actually physically associated with the nuclear matrix. DNA is methylated, and we have the presence of variant histones. So the question is, what is X exist doing to cause all of this? Well, the bottom line, so far at least, is it serves as a scaffold to bring all those deacetylases and repressive modeling complexes, etc., to the DNA along the X. In fact, more than 80 proteins have been found to bind the exist RNA at different places along its length. So the exist RNA is a gigantic delivery machine which takes all these protein complexes to where they need to go all over the length of the whole X chromosome. So here's a slide with a very intimidating list of some of the various proteins that have been found physically associated with exist, and they occur in different classes. And I'm going to summarize this on the next slide. Don't worry about all the gobbledygook on this one. Okay, so for example, CTCF and SMC proteins are architectural. They organize chromatin domains into loops, and we'll talk about that a little bit next time. We also have protein complexes involving chromatin remodeling, namely the SWI/SNF complex. Okay, this is named in yeast, but BRG is the same thing in human, but it's an ATP-dependent chromatin remodeling complex, and it can be involved in either activation or repression. It's able to move nucleosomes around. And of course, we have the proteins involved in histone modification, including PRC1 and PRC2, which contain subunits that ubiquitinate or install the repressive H3K27 mark. Okay, SPEN is a huge RNA binding protein that's associated with various histone deacetylases. So all the important players are here because we know what it takes to repress chromatin. We need to deacetylate, methylate it, and then compact it. Okay, so this is a representation of what exist looks like, and it's considered to be modular because it has a bunch of different regions that do very specific and very different things. And the ones that are known to be functional are labeled with letters, A, B, C, D, E, F, etc., to indicate the regions of the chromosome, or the region of the RNA, rather, that will carry the proteins that perform the various functions. Okay, so for example, a lot of these include tandem repeats. For example, repeat A consists of 7 to 8 repeats of a 26 nucleotide sequence. The other labeled regions also contain repeats that are characteristic of that particular region. So the bottom line is each particular region binds specific proteins whose job it is to install the various modifications along the X that are responsible for the inactivation process. Okay, so there have been about a gazillion experiments done on this system, and I'm just choosing one to illustrate kind of the general conceptual approach and just some of the results. Okay, so the idea is we will make deletions, first of all, in the exist. Loss of particular regions will lead to loss of specific proteins. Then we can look at the effects of loss of those proteins on ultimately what happens to the X as it's being inactivated. So we're not going to, in this experiment, identify the proteins that bind. We're just going to look at the effects of the processes, specifically on ubiquitination and methylation by polycomb repressor complexes 1 and 2. So this is just the tip of the iceberg in terms of the number of experiments that have been done to get this all figured out. Here's a picture of our mouse chromosome again with the X inactivation center. And at the bottom, we're showing the exist RNA with some of those regions indicated. So for example, A is implicated in silencing, the BC region in PRC recruitment, and E in localization, just for example. Okay, so for this, we're going to use a chip-seq experiment, just as an illustration of this approach that can be used for many, many different sorts of problems. Okay, so in this example, we will need an antibody that binds the protein of interest. Now, this could be a transcription factor. It could be any protein that binds DNA. It could be RNA polymerase. Okay, in this case, what we're interested in looking for is the ubiquitinated H2A or B, and the methylated lyse 27. And we're going to look to see if those marks are lost in the mutant form of the exist that is lacking the BC region. So in general, what we need to have for this is proteins that bind the region of interest, and we will pull down the protein with the antibody and then determine the DNA sequences where the binding occurred. Now, it seems kind of amazing that we could have a specific antibody that will bind the methylated form of histones and not the unmethylated form, or the ubiquitin form of the histones. But those specific antibodies do exist, and they allow us to distinguish nucleosomes that have those marks from nucleosomes that do not have them. Okay, so here's the overview of the ChIP-seq experiment in general. Okay, we start off with the DNA from a cell that's got protein bound to it. Now, that could be a transcription factor, or it could be a particular histone. Okay, the first thing we're going to do is cross-link the DNA to proteins, and that's not always necessary depending on how stable that particular interaction is. But generally, formaldehyde is used to cross-link the proteins to the DNA. Then, importantly, the DNA needs to be shredded up into small enough pieces that they can be distinguished from one another, typically 200 to 500 nucleotides, basically. And we're going to keep the proteins on them at that point. The next step is going to be to specifically immunoprecipitate those, and that's kind of the key step in this, where we have an antibody that will bind specifically to the protein that we're interested in this case, the methylated and ubiquitinated histones. Okay, now this pull down is not shown well on this particular slide because it's showing the antibody binding, but it's not clear why we would be able to then precipitate it. Well, in practice, what's done is it takes advantage of proteins A and G linked to either agarose or metal beads. Okay, what proteins A and G are proteins that bind to, in general, to the IgG class of antibodies. So those can be used to bind to the antibody that's bound to the protein and therefore allow us to centrifuge it out of solution by virtue of the fact that it's attached to a separose or metal bead. So that's the immune precipitation part of all this. So then, after we manage to precipitate those small regions of the genome we're interested in that are bound to the antibody, which is bound to the protein of interest, the DNA is then wash really well to get rid of everything else, and then release the DNA so that it can be sequenced. Now, this is done by generally now next generation type sequencing. So what's going to happen in next generation sequencing is we're going to massively sequence everything that's there, literally millions of sequencing reads. So we're going to get millions and millions of sequences, all very short. And then fortunately, there's very sophisticated software which enables those particular sequences to be sorted out from one another. So what happens is, of course, we know what the genome sequence is, and so the machine is able to take each individual sequence and then figure out where in the genome it came from. So you're going to get millions and millions of sequences which represent different parts of the genome where the binding to the individual protein was seen. So in this experiment, each sequencing read is located or placed onto some specific genome region according to its sequence, and that eventually, if you have an enrichment of a particular sequence at a particular location, you get a peak indicating that many, many reads were found at that location versus other locations where that protein is not found. So these are represented as peaks where the sequencing read depth or sequencing read number, the peak represents the large number of reads at that location versus reads elsewhere in the genome. So this is pretty much what I already said. Important thing being that you can pull down the antibody complexes because proteins A and G that are linked to something physically that you can centrifuge, namely Sephiros beads, so that allows them to be pulled down and collected, washed, and subsequently sequenced. So literally millions of bits are obtained, each of which represents a piece of DNA that was bound. So mapping the reads to the genome is very critical to this because each of those million reads is going to be has to be placed somewhere wherever it came from, and then these are represented and displayed according to where they came from and shown as peaks. So this will tell us where the modified histones were in the cells that we're looking for. Okay, so the bottom line is B and C repeats are necessary for recruitment of the two polycomb repressor complexes, PRC1 and PRC2, and subsequently, and that's what's shown in this experiment, that this is necessary for the formation of the ubiquitinated and methylated forms of the histones across the inactive X and not the active one. Okay, one thing to be careful of in this kind of experiment is these were not done with whole organisms. These were done with mouse ES cells that have been genetically manipulated to induce either the full-length exist or the mutant version according to the experimental needs. So this is not exactly a natural situation, but it's a very well manipulated one so that we can interpret what we see in terms of the modifications that are produced. Okay, so this was done by two different labs in slightly different ways, but the bottom line is in the absence of the BC region of exist, you get almost total absence of the silencing marks on the X. Okay, so in one experiment, we're going to compare the full-length FL version of exist to the delta BC region. Delta means deletion, so that deletion means that region of the exist RNA has been removed. And the antibodies, of course, are against H3 lys27 trimethyl and H2A 119 ubiquitin. So again, cells were lysed, exist was induced, cells were lysed, antibodies used, complexes pulled down, and then sequenced. Okay, so the bottom line is that the deposition of the repressive marks is virtually obliterated when the regions B and C are removed from the exist RNA. So I'm going to look at the slides with the actual data. Okay, these are a little hard to look at. The panels on the left are looking for the existence of the histone H3 modification, the methylation. The x-axis on these is location on the X chromosome. The other chromosomes aren't being shown. So what the peaks in each little spot represent is a location where the methyl antibody pulled down that sequence of the DNA. So the peaks tell us the DNA sequences with which those methyl groups were associated. And so you can see there's peaks and valleys all the way across the X in significant numbers. Okay, by contrast, if you do the same experiment and do see exist that lacks the B and C region, you see virtually no methylation of that particular histone across the length of the X. So in the absence of the B and C, the methylation did not occur, presumably because those complexes could not be delivered correctly to the X chromosome in the absence of that binding region. Okay, on the right, we've got histone, this is H2A, again, comparing the full-length exist to the one with the deletion. And again, the X chromosome is indicated by position along the X-axis. And we can see lots and lots of dots where that modification was observed. The antibody pulled down the modified histone, and the DNA associated with it matches that particular sequence on the chromosome. So the result is the same, that if you have your deletion of your BC region, you get very little, if any, formation of the ubiquitinated form of this histone. Another lab did the experiment in a slightly different way, but got the same result. Okay, in this case, we're going to be comparing, slightly differently looking. The top on each slide, the top panels are the methylation. The bottom panels are the ubiquitination that we're looking for. So it's two separate experiments being represented here, really. Okay, so again, we induce the expression of the exist RNA, and we look in wild-type cells first for comparison. So we can see that the future inactive X is coded with the correct modification as expected all along its length. There are a lot of reads all the way across the X with a few little gaps, indicating that those methylated histones were present at nucleosomes all along the length of the X. The active X, however, does not have the repressive marks, as you would expect, that there was very little sequence read from the active X in response to the wild type, as expected, because it's not repressed. Okay, by contrast, if we look at the delta rep B, which is to say the delta deleted form of the repeat in the B region, we see that the inactive X does not have the methyl marks, as should be expected for repression, and of course the active X doesn't have it either. So the result here is that the lack of the B region of the exist obliterated or caused a total failure in the ability of the exist RNA to deliver those repressive complexes to the correct locations on the X. Okay, the same is true on the bottom panels, but we're looking at ubiquitin instead. Again, the XI in the wild type becomes nicely coated with the ubiquitin form of this histone, and it exists at locations all the way across the length of the chromosome. The active X, by contrast, does not have this particular modification, as expected, since it's a repressive mark in that chromosome. Okay, the experimental form, again, is we are deleting region B from this particular version of exist, and as a result of this fact, we do not get any precipitation of ubiquitinated histones as a result of this modification. So we can't ubiquitinate our histones in the Rep B mutant because the protein is not able to bind there because of the absence of the binding site. And as a result, the complex is not delivered across the X as it should be, and you don't get the repressive mark. The X active, of course, isn't repressed, so that doesn't have any repressive mark, which makes sense. Okay, so here's a way of visualizing what's happening. The full-length exist binds the complexes for this particular modification in the regions B and C, mostly B, and as a result, it's able to deliver those remodeling complexes or modifying complexes to the chromosome all along its length, with the result that you get buildup of the repressive marks along the length of the chromosome. In the absence of the BC region, those marks are not formed because the protein complexes can't bind because their binding sites are missing from the exist. As a result, you're not able to repress. You're not able to establish repressive marks across the length of the X. Okay, don't stress too much about this, but this is another example of a fish experiment. Only, in this case, it's more like a Western blot on a slide, because what we're doing is mostly we're looking for proteins. So what we're going to do is use antibodies to the specific proteins, and then we will have a fluorescently labeled secondary antibody, which will allow us to determine where the protein is located. So this is like the standard fish where we just have the chromosomes on the slide and stain them with DAPI or stain with nucleic acid probes, but in this time we're adding on the ability to actually locate a protein by means of an antibody, which is fluorescently labeled to the particular protein of interest. Okay, so what we're going to look at mainly is this protein called EZH2, which is a component of the PRC2. Okay, and this we're going to see that it co-localizes with the exist RNA that we can see in the fish experiment. So the background color is blue, DNA stained with DAPI. The exist RNA is colored pink, and this was obtained by hybridizing it to labeled DNA probes that are complementary to different parts of it. And then EZH2, we have a primary antibody to that protein, and the secondary antibody is labeled with a fluorometric fluorometric compound, which fluoresces green. And it makes some astonishing images, really. Okay, so what this shows, and don't worry about all the extra pictures, we're going to look at the column on the left, where we have a silencing control, and we're looking at the presence of the exist RNA as something that stains kind of pinky red along one edge of the nucleus, as you can see. You can see the background is blue, which just represents the DNA. And then importantly, we have the antibody staining of EZH2, which is a protein component of a repressive complex, and that is stained green. So we can see by looking at it that in these wild-type cells, the exist RNA and the EZH2 protein are found in the same regions of the nucleus, which is to say that they co-localize, which is what you'd expect if the EZH2 is part of a chromatin modifying complex whose job it is to install these repressive marks. Now, just for illustration purposes, on the right-hand column, we have 3 cells in which the HDAC3, which is a histone deacetylase, has been silenced. We see the exist RNA as in pinky red, but then we look where the EZH2 is, and there's a conspicuous absence of EZH2 from that region of the nucleus where the exist is localized. So there is no localization of EZH2 in the absence of this particular histone deacetylase. And you can see that in the merge still looks pink. It doesn't look yellowy like it would if you put the two together in a merge. So that just illustrates this general thing. So this is like a Western blot on a slide, because with the EZH2 is actually detected with an antibody rather than with a nucleic acid probe, as you would typically see. Okay, so again, we're going to treat this slide like a Western blot. We're going to float the antibody, rinse, use a secondary antibody, which is labeled and that with a color, and so that enables us to see them both. So this is an example of co-localization, or lack thereof, in the different cells. So I think this is what I said. So the merge superimposes the image, and we can see very clearly that the EZH2 protein is found along in the same place as the exist cloud in the wild-type cell. So you can look at the rest of it. The HDAC3 is clearly necessary for the correct localization of EZH2, etc.